Hello and welcome. We've put together a video showing how we extract DNA from tissue samples. We receive these tissue samples preserved in ethanol from trappers and research managers in Maine. We'd like to say a special thank you to everyone that has collected tissue samples for us so far. The first step here is to take a small piece of those tissues as input for the extraction. We're looking for about 25 milligrams of tissue to cut up into smaller pieces to increase the surface area the enzymes and chemicals have to work on for digestion. This sample is T1313, a muscle sample from a beaver harvested by Pleasant River near Bethel, Maine. The subsample for T1313 weighed 25.1 milligrams, so we were right on the money for this one. After subsampling, we add buffer ATO and an enzyme, proteinase K, to digest the tissues. The DNA right now is held within cells and the nucleus. Proteinase K will digest proteins and help get those DNA strands out in solution where we want them. Here, we're using a vortexer to mix everything together, and then a centrifuge to get everything back down to the bottom of the tube. So this is what the samples look like now, and we'll leave them to digest overnight or for a couple days at 56 degrees Celsius, which is the optimal temperature for proteinase K. And of course we have to give them a little pat for good luck. So after digestion, as you can see, the visible tissue has been digested and the DNA is in the solution. Now our goal is to separate that DNA from all of the digested junk that's also in solution, and to do this we do a column purification. The first step for the purification is to add a buffer that will help the DNA bind to a column made of silica that the sample will flow through. The DNA will bind and stay on the column while everything else flows through and gets discarded. Here is the column, and that white part is what the DNA binds to. Here I'm adding the sample solution to the column. It requires force to make the liquid move through the column, so we'll use a centrifuge to apply that downward force. Now that the DNA is bound to the column, we'll discard the liquid and wash the column twice to get rid of everything that is not DNA. After washing the column, we transfer the column to a final tube that the DNA will be collected in. To get the purified DNA off the column and into solution so we can use it, we add a buffer that disrupts the bond between the column and the DNA. We'll use the centrifuge again to force the elution buffer and DNA through the column and into the collection tube. So during this process, we went from the DNA being in the tissue to purified DNA in solution. Thank you for watching, and the next step for these samples is to prepare them into what we call libraries. This will prepare them for sequencing.